Thank you very much for this invitation. I'm very happy to speak today about how circulating metabolites could help us in differentiating patients with ischemic stroke from stroke mimics. My name is Steffen Tiedt. I'm a clinician scientist and junior research group leader at the Institute for Stroke and Dementia Research, and thanks again for this invitation. Our work was motivated by the fact that in the emergency department, we're often confronted with the diagnostic challenge how to differentiate patients with ischemic stroke and stroke mimics, so such as epileptic seizures or migraines, because they often present with the same symptoms. And misclassification of these patient groups may result in inappropriate interventions, withholding treatments with proven efficacy, such as thrombolysis, and inefficient allocation of resources. The diagnostic gold standard is the multimodal CT, but has insufficient sensitivity to differentiate these two diagnostic entities. So obviously, as you all know, the circulating metabolome may capture pathophysiological events in multiple organs, including the processes related to acute ischemic stroke. And thus we set out to identify a set of circulating metabolites that could help us differentiate these two groups. To do that, we sampled serum upon hospital arrival, so directly when the patient would come into the emergency department, before any imaging, before any medication, across 5 different samples of patients here with ischemic stroke. And in the first 2 sets, neurologically normal controls and also stroke mimics, so the disease or the control group of interest. And overall, this comprised almost 1,000 patients. We used an untargeted metabolomics screen here to identify in the first set almost 700 metabolites across the different classes, and here distributed across the different pathway levels here as an example for amino acids. We only analyzed some metabolites that would be detectable in more than 50% of our samples, and using 2 different linear regression models adjusting for different sets of confounders such as comorbidities, obviously also age, but also glomerular filtration rate, we identified 49 different metabolites as being different between these 2 groups in the first set, so neurologically normal controls and ischemic stroke patients. From these 49 metabolites, we validated in an independent sample using the same platform 41 in this different second set. We restricted this 41 metabolites to only endogenous metabolites, that would be 30 then, and we looked whether we could differentiate these 2 groups, so ischemic stroke patients from neurologically normal controls using a random forest regression, and that was basically a very nice AUC that we got here. But the next question was whether that would only rely on the machine learning method, but that was not the case. Also, other machine learning techniques such as support vector machines, k-nearest neighbors, performed similarly well. We next asked whether these 30 metabolites would indeed be all giving us additional information, and first looked whether there's any collinearity between metabolites, because that's obviously known that that is sometimes the case. And indeed, we found that there was significant collinearity here between mostly fatty acids, as you see here in this heat map ordered by hierarchical clustering. So we wondered, how can we identify the most important metabolites for our diagnostic questions? So differentiating ischemic stroke patients from stroke mimics. And we here used a variable importance analysis using random forest regression, and they identified ADMA, so asymmetric dimethyladenine, symmetric dimethyladenine, pregnenolone, sulfate, and adenosine to be the most important predictors, or let's say parameters in this model. So we restricted the model to these 4 metabolites, and indeed, again, we got on an AUC that was obviously lower than the initial one that we got, but still significantly better than the diagnostic gold standard, the multimodal CT, so computed tomography here, that would be usually done in the emergency department to differentiate these 2 groups. That is our derivation sample, so set 3 of our analysis. We also did this in set 4, so an independent group again of 105 ischemic stroke patients, 105 stroke mimics, that was matched using propensity score matching to adjust for additional confounders or to control for additional confounders, also such as medication. Again here, same result, metabolites perform much better than computed tomography. And we were then asked again by reviewers to perform an additional replication sample. This comprised now here 500 consecutively recruited patients, where we basically see again the same result. Metabolites perform better than CT, but in this group, compared to the previous sets, we also found that computed tomography would slightly add to the AUC, to the ROC curve. We performed sensitivity analysis for patients that would be treated with thrombolysis, and we could, basically excluding some of these patients for different reasons, but that gave us the same result basically. And we also showed that these 4 metabolites could differentiate between ischemic stroke patients and neurologically normal controls, which you would also kind of expect. To characterize these metabolites, we profiled them longitudinally, and we found that the acute phase here is increased. So indeed linked to the acute phase, different etiologies do not bring different values except for symmetric dimethylarginine. And we have basically, they're mostly independent of stroke severity, and sampling time does not only for adenosine. We did a systematic review showing that basically all these metabolites that our set would perform significantly better than previously published sets, and our paper has been published in the Annals of Neurology this year, and is obviously, you can have a look at it to find more details. With this, I want to thank our collaborators, mostly Christian Giger and Edgar Schwedham in Hamburg and here in Munich. Thank you for your attention.